This is a brief lecture on the physiology of the arterial tree. It explains why we need to freshen up old theories that reach back far into the 19th century. In particular, it justifies why we should embrace the recently formulated theory of arterial acceleration. In 1628, Harvey was the first to realize that blood circulates through the body. Up to that time, it was believed, based on Galenic tradition, that venous and arterial blood flow were different, the latter being produced in the heart and distributed over the body tissues, whereas venous blood was supposedly produced by the liver. Harvey also understood that heart contraction was the energy source behind blood circulation. Harvey realized that blood circulated through the body and that the heart was the energy source, bringing it into motion. The heart is a volume pump. By injecting blood into the aorta, the pressure is increased, relieving itself via the many branches, arteries and arterioles to all capillaries within the body. Let us now consider the arterial tree. There is an exponential increase in number of arteries and arterioles to its periphery. The more distal an artery, the smaller its diameter and the shorter its length. The structure of arteries changes to its periphery, the wall becoming thicker compared to the lumen the more distal we go. At the far end are the capillaries, with a diameter of several micrometers, allowing the passage of individual erythrocytes and white blood cells. Capillaries are crucial for the exchange of nutrients and waste products. They ensure local homeostasis. Any signs of ischemia will be washed away and transported to the control centers of cardiovascular regulation. For the sake of the argument, let us perceive the arterial tree as a set of rigid tubes. What would happen with a drop of blood entering the aorta on the left side of this figure? Since the rigid tubes are unable to expand and fluid is non-compressible, any volume entering the aorta on the left will cause an identical amount of fluid to be released from any one or more of the arterioles towards the right. The increase in pressure caused by volume entering at the left will disappear instantaneously with volume released at the right. The energy required to enter blood at the left will be no larger or smaller than required for outflow at the right. Just for emphasis, with rigid tubes, pressure curves and flow curves are synchronous. In the model for cardiovascular simulation, this situation can be approached by greatly reducing the capacitances in the systemic arterial tree. The flow curves become more cubic and more similar to the input flow, unrealistically modeled as a block pulse. In 1809, Young reported on laboratory investigation he had performed measuring the resistance of tubes of various diameters. He stated that the resistance of a tube would increase the narrower its lumen. In agreement with Harvey, he was convinced that the arteries themselves do not add to the energy of heart contraction. Based upon the observations by Young, the theory of arterial stiffness was established, stating that arterial resistance becomes larger in arteries more remote. However, the assumed tapering of arterial branches ignores the effect of exponential branching on total cross-sectional area of the arterial tree. In fact, total cross-sectional area of the arterial system increases manifold on its way to periphery. In a way, the cardiovascular system can be seen as a funnel. Pulsating energy delivered through the small cross-sectional area of the aorta is expected to dilute along the branches of the arterial tree. What this means energetically is symbolized by this young boy trying to extinguish a candle by blowing through the narrow end of a funnel. For instance, this is a table with estimated dimension of the arterial tree. See how the cross-sectional area increases to its periphery from 4 squared centimeters at the aorta to 600 squared centimeters at the level of the capillaries. To 6,000 squared centimeters at the level of the capillaries. We have added an extra column to the table shown earlier, the estimated volume per subgroup of arteries. When the length of an arterial branch is the reciprocal of the total number, the volume contained by every subgroup of arteries will remain almost identical. Any volume entering in the aorta of a rigid arterial tree will cause movement of a similar amount of volume within every subgroup of branches. But when we look closer to an example of an arterial tree that is asymmetrical, we come up with new conclusions. For a single drop of blood to reach the far end of this tapering arterial tree, the total resistance increases with every tiny branch added to the tree, since these are resistances in series. But towards the periphery, the arterial tree splits up in many branches. At every node where a single artery splits into two, the resistance of each individual artery increases, but the overall resistance of two arteries in parallel becomes smaller than of either of its components alone, causing the resistance met by a droplet of blood to enter a group of arterioles to drop with every branch added. As for a subtree, the resistance becomes smaller and smaller. And in an asymmetrical tree, this holds true as well. Still, the resistance will increase along the arterial tree. There are differences in total resistance for arterial subtrees close to the heart as compared to subtrees more distal. This has to do with the length that the blood has to travel. So if we enter 100 drops of blood in this asymmetrical arterial tree, the blood will distribute unevenly over all subtrees. Subtrees close to the heart will receive more blood than those more remote. And the heart has no powers to direct blood preferentially along the various parts of the arterial tree. So it may be no surprise that the brain has been positioned close to the heart. The brain is the most demanding and energy-consuming tissue in the human body. So it requires a low resistance to blood flow. Distant parts of the body require less. Now, instead of rigid, arteries are generally believed to be viscoelastic. This allows pulsatile energy from the heart to be stored in the most proximal part of the arterial tree. In a balanced system, the sum volume pumped into the aorta will also be relieved in the system's capillaries. But a time shift has now occurred. Pressure curves and flow curves are no longer synchronous, and the shape of the pressure wave will vary along the arterial tree. It led Frank to propose his Windkessel model in 1899. The heart is responsible for pumping blood into the aorta. The elastance of the large arteries allows the blood volume to be kept at high pressure, provided that peripheral resistance is high enough to prevent a massive outflow. And the resistance should in fact be high in every branch of the arterial tree, so that the arterial pressure can be maintained and does not leak away gradually, allowing the heart time to replace whatever volume is released. At the end of the 19th century, Greshey and von Kries postulated that waveform characteristics in proximal recordings of the arterial pressure wave were the result of forward and backward traveling waves. This hypothesis was inspired by work of von Helmholtz, who studied sound waves in closed and partially open tubes. The high outflow resistance causes a reflection of the antegrade pressure wave from the heart. It goes without saying that these recurrent waves oppose the natural flow of blood and cause energy loss to the pressure wave from the heart. What shall this mean for the waveform? The recurrent waves are thought to cause an indentation in the flow velocity wave, varying in time from proximal to distal branches. In proximal branches, these recurrent waves are thought to cause indentations in the systolic waveform. In pressure recordings, recurrent waves are thought to cause augmentation, even during the systolic part of the recording when points of reflection are close to the points of measurement. So the energy generated by heart contraction is expected to dilute along the branches of the arterial tree, lose impetus by the Windkessel phenomenon, favor proximal subtrees with respect to those more distal, and encounter wave reflections that are flow opposing. And then we didn't mention blood viscosity and the effect of gravitational force that will also undermine an effective transformation of heart contraction into blood distribution. Based upon a number of observations, in 2014 the theory of arterial acceleration was postulated. This theory proposes that every heartbeat initiates a peristaltic wave of contraction in the smooth muscle layers of the arterial tree, spreading from proximal to distal branches. In the arterial wall, smooth muscle cells are arranged circularly. Smooth muscle cells are known to be stretch responsive, so a sudden increase in intraluminal pressure will cause depolarization of smooth muscle cells. Individual smooth muscle cells are connected to each other by gap junctions. Theoretically, this will cause a local depolarization to spread along the length of the arterial tree. Calcium sparks have been observed in smooth muscle cells. On a cellular level, these short bursts of self-limiting calcium inflow may underlie the arterial acceleration observed in the arterial system as a whole. It still needs to be demonstrated whether these calcium sparks are able to give rise to a brief and rapid contraction within the smooth muscle syncytium. Arterial acceleration enhances blood flow. Since it is short-lasting, only active during the first instances of every heart contraction, it spreads more rapidly along the arterial tree than the blood volume, which is delayed by the Windkessel phenomenon. Pressure is augmented during the first instances of every heartbeat as flow is enhanced. Where the recurrent waves in arterial stiffness are flow opposing, arterial acceleration enhances flow and adds energy to the pressure wave from the heart. The effect of arterial acceleration can be demonstrated in the cardiovascular model. It increases pulsatility at the remote ends of the arterial tree. Arterial acceleration helps to overcome the resistance of small arteries, bringing the blood into motion even when tissue conditions are less favorable, for instance due to external pressure. The effect of arterial acceleration was demonstrated in a simple experiment. Unfavorable tissue conditions were simulated by inflating a blood pressure cuff placed around the hand. A Doppler probe was used to measure the velocity of blood flowing into the tissues of the hand. The Cis1 component prevails despite unfavorable tissue conditions, whereas the Cis2 component and diastolic flow are completely absent during cuff inflation. After cuff deflation, these return in the flow signal and are temporarily increased during the period of hyperemia. So we conclude that the physiological origin of the Cis1 component is different from the Cis2. It makes sense to distinguish both components in pulsatile signals and study their different behavior during, for instance, pathological conditions on the ICU, such as sepsis, hypertensive crisis, and/or intracranial pressure elevation. I thank you for your attention, and we will proceed with the second part of this lecture that will deal with different pathophysiological situations.